Okay, we're back with Chapter 9, Part 2.2. So in Part 2.2, we're going to focus mainly on the topic of mutation. So in Part 2.1, we talked about how the sequence of the DNA ultimately leads to RNA that codes for how to make protein. And the important thing to think about moving into this next part is that proteins will ultimately dictate the functions and the characteristics of cells. So any change in the coding can potentially lead to a change in the outcome of protein function. This could lead to a variety of differences in the final product. But mutation is defined here as any permanent change in the DNA sequence of a gene. And there's different ways to define that. It's not a definition that's set in stone, but that's the idea. It's a change in that sequence. So if the sequence gives us the original protein, any deviation from that is considered a mutation. Now, mutations can occur for two general reasons. One reason, and this is actually the most common, is called a spontaneous mutation. And during DNA replication, any error made is considered spontaneous. So our DNA has to be copied at a very vast rate, many times, over and over and over, trillions of cells, each of them dividing for the most part. And at some point, the replicating enzyme, DNA polymerase, makes mistakes. And these mistakes are referred to as spontaneous. There's no exact reason for them. They're random. There's probably some genetic element to them, ironically. In other words, some cells and some people are more susceptible to these types of mutations than others. But otherwise, there's no real reason for them. So they're called spontaneous. Other types of mutations are referred to as induced mutations. These are the kind of mutations that result from some sort of exposure in the environment, damage from a variety of different sources. The source itself is often referred to as a mutagen. In other words, something that can cause mutation. Think of these as environmental in their nature. Typically, something around you you're being exposed to is causing an induced. These could be things like toxic waste, X-rays, even UV light is potentially mutagenic. This is one of the issues you can run into if you go to a tanning bed. You're putting yourself in front of a high source of a mutagenic physical material, which is UV light. So cigarette smoke is another example. These are all things that fall into the induced category. So something in your environment causes that mutation. Now, ultimately here, we want to focus more on what happens because of the mutation as opposed to what causes the mutation. I think the cause of mutation could be a very different focal point, and so here we're looking at what's the actual outcome of mutation. So ultimately, as I said before, the normal gene should produce a normal protein, and a mutation, at least potentially, can cause either an abnormal or, in some cases, no protein at all. Another way to think about this is a change in the genotype can result in a change in the phenotype, because phenotypes are often the result of the protein or RNA sequence itself. Now, the result of this type of mutation, or the result in the change in the phenotype, can come up or show up or manifest in a wide variety of ways. So here we're going to sort of bullet point a handful of generic ways that this could happen. Number one, in some of the most obvious types of mutations, are they change the physical look or the shape, size, color, etc. of something. More abstract, beneath the surface, are biochemical changes. These could be things that occur in our digestive enzymes, the way our body absorbs or manages vitamins. The difference is in the rate of metabolism. All these things can be subtly affected by changes in the proteins that make up our biochemistry. Genetic control mechanisms. Every cell in the body has a mechanism for turning on or turning off protein expression or cell growth, or a variety of different mechanisms for feedback. Any mutation in these mechanisms can lead to a failure in the regulation of cell growth. This, by the way, is how virtually all cancers arise. Cancer is a group of cells that are growing at an uncontrolled rate, and these always occur due to mutations in the genetic control mechanisms themselves. It's like a little switch that a cell has that turns it on or turns it off in reference to growth. Cancer cells are essentially stuck in the on position, and they lose the ability to stop their growth whenever it would normally occur. And these are, in general, mutations in genetic control mechanisms. Another broad category, this is especially seen in bacteria, resistance to chemicals, and we see this with antibiotic resistance. Antibiotic resistance ultimately is a mutation that makes a microbe change its interaction with a chemical, like an antibiotic. And so in these instances, they gain resistance in reference to that mutation. So, as I said, many more specific instances of a mutation can occur, but this gives you a pretty good idea, a pretty broad overview of what that change in the phenotype might mean. Now, if you actually think about mutation, you would sort of assume anyway, if you were to stop and think, that mutations would probably always be bad, and change would always result in some negative outcome. And while that is more likely true than not, the resulting mutation can actually occur in one of three different outcomes. So they can, in fact, be harmful. But they can also be neutral, meaning that the effect isn't really obvious. It may not even be noticeable. And in some rare cases, a mutation can actually be beneficial. I think of a beneficial mutation in probability sense, sort of like a lottery ticket. The odds of a mutation happening in a way that is beneficial is very low, but possible. Kind of like winning a lottery ticket. The odds are not in your favor, but they can happen. And so beneficial mutations, I think, are sort of in that general ballpark of probability. Not likely, but possible. So it can occur. Now, the truth is, when it comes down to whether a mutation is harmful or neutral or beneficial, ultimately it comes down to what exactly is being mutated. So the next thing to recognize is that proteins in the body are sort of like organs and limbs and body structures in our own bodies, is a way to think about it. Some proteins are critical. And in that sense, think of like a protein as like your heart. Your heart is critical to your overall body function. Some proteins in the cells are critical to cell function. And so just like if something were to happen to your heart where it would stop functioning, it would immediately be critical to your overall survival. The same is true for cells. Certain proteins inside the cells cannot be mutated virtually at all. Any significant change will almost immediately result in cell death. You might see this, for example, in proteins that process oxygen. Vital proteins for oxygen metabolism, for oxygen-breathing organisms like you and I, are critical. Very, very little room for mutation in those. But other proteins that may play a role in cell surface structure or the cytoskeleton may not be as critical. So slight changes may result in different cell shapes or different structures in the cell that may or may not be critical. So overall, there are some proteins with a little room for fluctuation and other proteins that are absolutely a staple, a stalwart, so to speak, in our cell structure that are not really able to be mutated at all. So these types of proteins that are vital, if you have mutations, almost always harmful. So here's an example. The human insulin gene is critical as it codes for an essential protein, which is insulin. Insulin, as many of you probably already know, helps us regulate our blood sugar. If we were to have a mutation to the insulin protein, the gene that actually makes insulin, we would be unable to regulate blood sugar on our own. And this would be devastating to our body's physiology. Now, we know that we can take drugs and we can take supplements to offset that, but on a base level, as far as our body's ability to stand on its own, This is what I would consider more of a vital function, and a mutation here would disrupt that vital functionality. Now, there's other genes in the body. For example, the AR gene, which stands for an androgen receptor gene. This is a gene that, in a sort of a complex roundabout way, ultimately can control the amount of body hair we have as humans. And body hair is not really critical to our survival, especially since as humans, we're able to coat ourselves and warm ourselves. But ultimately what this gene can do, in the mutation of this gene, it can actually cause hair follicles to be larger or smaller and more pronounced or less pronounced. So while no matter what we do, we need insulin regulation because we have to eat and we have to consume foods that require insulin to help regulate that blood sugar. Our ability to survive with more or less body hair is much more neutral. It may be good, but it's not critical. So genes to something like a body hair gene would be more in the neutral category, where genes to the human insulin would be more in the harmful category. And often, and the reason I use this as an example here is because, especially with hair, is because oftentimes what makes a gene mutation neutral or harmful or even beneficial has to do with the environment. So you and I as humans, we can easily control our body temperature by adding extra layers of clothing. And this goes all the way back to the prehistoric humans with animal furs and still used in some cultures today. But we can artificially warm ourselves. Take that same kind of mutation, though, and apply that to an animal that requires that for survival, like, for example, a mouse. And that same mutation could easily be an absolute dead end for that particular rodent. So the same mutation in one species could essentially be neutral and then harmful in another. And then it's possible, by the way, that could even be beneficial in some cases, where if you put the mouse in a very warm environment, having less hair might allow it to cool itself better. And that would probably not be typical or expected, but it's possible. So ultimately, what I'm saying here is that the ultimate outcome of a mutation, in some cases, is actually determined by the environment. And these are often in those in those genes that are not critical for our survival. So, it's almost impossible to draw up a scenario where a gene involving oxygen metabolism, it's almost impossible to draw up a scenario where that's beneficial or neutral, because oxygen is a minimal body function. But when it comes to hair, we can see how it may be neutral or harmful, or even in some rare cases beneficial, depending on what the environment around that organism is like. And that's one of the things I want to get to next here a little bit more, is about how mutation and the environment go hand in hand. So there's a really famous study in biology that occurs with an organism called the peppered moth. And the peppered moth was studied around the 19th century, and spent a lot of time, field biologists spent quite a bit of time studying this insect. It was very common. This was in England around the 19th century. It was very common, so it was well known to biologists. But it also was being recognized to have been impacted by human pollution and some of the effects that the human population was having on the environment as a whole. So they were studying the moth as a way of looking at human impacts, in addition to just studying the moth itself for the sake of science. But the name of the peppered moth comes from its peppered appearance here, as you can see. Now, if you were to go out around this time frame, and I think as of even today, you would see that the majority of peppered moths look exactly like you see here, hence the name. So some moths in the population, small percentage typically, exist in what's referred to as a mutated phenotype, which is, in this particular instance, a dark color. So a change in the phenotype in this particular case results in a dark coloration. Now, normally the ratio in the environment would be very low. So the mutant moths can exist at a low level, and we're sort of taking out some of the discussion about how these mutations occur. So these mutations are genetic, they are a change, but they're passed down in a hereditary standpoint. So some moths in the population are always going to have that dark phenotype, whereas the majority are going to have the speckled colored, peppered look that you see there. So we're sort of bypassing a deeper discussion on how exactly these types of mutations arise, but we're just sort of saying, for the sake of the environmental aspect here, that the two phenotypes occur, the mutant and the normal. And the mutant is in a very low percentage compared to the normal under most circumstances. Okay, that was at least the case for the peppered moth around the 19th century in England. And what happened over time, well, let me back up, sorry here. The reason why the mutant moth was not ever growing much in population, it wasn't actually the fact that the mutant itself had any serious issues. The mutant, in other words, could reproduce, it had the ability to fly, it had the ability to mate and eat, and all the normal things that the moth would do. The main issue, though, was just the color. That was really the main problem for the moth. Now, the reason why this specifically was a problem, because the primary predators for these moths are birds. And birds are visually orientated. In other words, they spot their prey visually. If it was smell, or if it was motion, or if it was some other sense, it may not have had much of an impact. But because it was the color itself, the birds were able to spot the mutants better. Now, even more specifically, the peppered moth, in the normal environment where these moths are found, could easily blend in to the trees in that environment. And so when they were in their normal habitat, you can see the wild type here, the normal colored peppered moth, it would blend in fairly well. And the trees themselves acted as a natural source of camouflage. And under those same circumstances, the mutant moth stood out, and it didn't have the same level of camouflage. And so the idea was that the birds were visually orientated, and therefore the dark moth, under this set of circumstances, was the easier prey of the two. And so this is why scientists hypothesized that the normal color resulted in a higher population, because it actually gave them a survival advantage under these circumstances. Whereas the mutant could survive, it never was eliminated entirely, but it didn't survive at the same rate, so they were harder to find. Now, fast forward in time, and this study was ongoing over several decades, and what they've started to notice was that in the same areas the peppered moth existed, lots of pollution started to enter into the environment. This was around the turn of the Industrial Revolution. There was lots of soot and smoke, and not only did the soot actually artificially darken the trees, but it also killed off some of the natural, what are called lichens, that grow on the trees that also contribute to that light color. So it was an actual darkening, but also more of an alteration in the symbiotic organisms that grew on the tree that helped contribute to the color of the tree. And so in a variety of ways, the pollution around that time actually started to change the color of the environment where the moths were living. And this is one of the things that really drove the scientists to go out and study the environment, was to see what was happening. Now, this effect today is referred to as industrial melanism. Ultimately, it's the darkening of pigments in organisms exposed to industrial pollution. That doesn't mean that the pollution directly causes melanism. It just means that it can affect them in a way that drives a change in color. So in this case, the moths didn't get darker because of the pollution, but the pollution drove a favorability towards the moth, the darker moth. So as you can see here, once the environment changed due to the industrial melanism and the pollution in the environment, what you actually saw was that many of the normal moths had a harder time blending in, because on those same trees in that same environment, suddenly their phenotype was no longer favored because of the change in the environment itself. And so over time, the dark moth was suddenly had an increased survival advantage, started to grow in population. And scientists actually measured an increase in the mutant moth in reference to a decrease in the normal moth. And so this was something that sparked lots of attention, and it brought a rise in the awareness of the pollution at the time. And one of the many things that sort of brought about a change in environmental awareness and an increase in our recognition that humans were actually having a role in the alteration of species in the environment over a short period of time. By the way, this was only over a couple of decades that this was measured. Interestingly, over time, the pollution went down, the environment went back to its normal scenario, and the moth populations actually flipped back into the normal ratio. So this was an interesting example that showed just how much the environment can have an impact. And so that's the main takeaway here. This is just one example, and an example, by the way, that's obvious to the people who studied it and based upon the outcomes that they showed here. It clearly demonstrates that there's a relationship between the favorability of a particular phenotype and the particular environment that they're in at that given moment. It also shows us that because an organism's environment is subject to change, the outcome of mutation may be neutral or beneficial, depending on the circumstances. So this is where it's important to recognize from a big picture that mutation itself can actually be harmful or beneficial or neutral, actually depending on the environment around them. Now, one of the last things I want to get into here are how the mutations themselves occur in the DNA. So there's one major type of mutation that occurs, which is called a point mutation. So a point mutation is a mutation that affects only a single DNA nucleotide. So in all the letters of our genetic alphabet, in all the sequences, only one letter is affected. And this is referred to as a point mutation. Now, a point mutation can occur in three specific ways. Number one is called a substitution. So this is where the original nucleotide of one position is altered into a different nucleotide. Additive is where we end up throwing an extra nucleotide into the sequence. And deletion is where we remove a nucleotide from the sequence. And as we'll see going forward here, additive and deletion mutations tend to be the most harmful. But all three of these fall into the category of a point mutation. Now, what happens as a result of any of those three can result in four different scenarios. And we'll go through each of these here. But missense, silent, nonsense, and frame shift. So let's take a look at a missense mutation. A missense mutation is defined as any mutation that changes an amino acid in the primary protein sequence. So that's the takeaway. The idea is a change in the sequence through transcription and translation ultimately leads to a change in the protein sequence itself. So here's an example showing the DNA sequence, the RNA sequence that's being transcribed, and then the resulting protein chain, the amino acid chain that would occur during translation. If we have a change here, a point mutation substituting CT and the G here into CT and a T instead, now our transcript will read GAA, and instead of the amino acid ASP, we'll have the amino acid GLU. This is aspartamic acid, and this is aspartic acid and glutamine is what these stand for here. So the point is that we end up changing the actual protein sequence, and so the term here is called a missense mutation. Now, what actually results of that is not necessarily implied. A missense mutation could be very harmful, it could be very neutral, or it could be beneficial. It doesn't mean it has to be good, bad, or neutral. It just means that you actually have a change in the amino acid sequence. Amino acids themselves are also variable in their importance. Some amino acids in the chain are more or less irrelevant. They don't necessarily play a big role in the overall folding of the protein. Other amino acids are critical, and changing that one amino acid could change the overall structure of the protein. So it's only implied that the change occurs. It's not implied how significant that change is. Now, the next type is called a silent mutation. This is where we actually have a change in the sequence, but a change in the DNA sequence, but no change in the amino acid sequence. And the only reason this is possible is due to what's called degeneracy in the genetic code. And I'll show you the chart here in a second, but it turns out that many codons that code for amino acid have multiple codons coding for the same amino acid. And this is what's referred to by degeneracy. We have a handful of codons in some cases, all coding for the same amino acid. So in the case where we change from one codon to the other, both of which code for the same amino acid, we have a technical mutation. We've technically changed the genetic code, but we haven't actually changed the amino acid. And so the change is recognized by calling it a silent mutation. So we recognize that there is a mutation, but we call it silent because it doesn't actually change anything in the amino acid sequence. So for the most part, they're kind of irrelevant in terms of their impact on the protein itself. So these will not result in any significant impact. So here you can see, for example, CTG and the CTA mutation here with the G to A doesn't change the amino acid. And if we look on the next slide here, this is our codon chart I showed you in the last part there. And if we look where aspartic acid is coded, GAU and GAC both code for aspartic acid. So if we back up here, the codon, which is the RNA sequence we're showing here in the middle, in the original is GAC, and then the mutation is GAU, both of which code for aspartic acid. So we have a mutation, but we end up with the same amino acid. That's called a silent mutation. The next one here tend to be on the more lethal side of things. And this one here is called a nonsense mutation. And the idea is that it results in a protein that is generally nonsense in reference to what you had to start with. So typically we're implying a massive nonsensical change in the outcome. But what happens here is that we will change a normal codon into a stop codon. And anywhere we change a normal codon into a stop codon, protein termination stops at that point. And so we will take what is normally a long protein chain and terminate that short whenever this type of mutation occurs. And anytime you cut a protein short, you're going to almost always result in a non-functional protein. It's very, very, very unlikely that that's going to result in anything resembling the original protein functionality. So these tend to be what are referred to as a lethal mutation, very, very serious in their outcome. Now, how this is even possible, I'll go back to this codon chart here. Take an example like tyrosine, one of the amino acid coding codons. Tyrosine has the sequence UAC. If a mutation changes this third position here from a C to an A, instead of a codon for amino acid, we now have a codon for the stop. And that could occur in UAA or UAG. Either one of those are similar to tyrosine, but instead code for a stop message instead of the amino acid. So anywhere those occur in the chain, that will immediately stop the protein short of replicating or translating to the full termination point. So similar ones happen with cysteine. Cysteine UGC could code to UGA, which would also result in a stop sequence. So because we have amino acid codons that are very similar, it only takes one little letter in the right position or the wrong position, depending on how you look at it, to result in a nonsense mutation. The last type here is called a frame shift mutation. Frame shift mutations are a little more difficult to immediately wrap your head around, but you have to recognize that a gene consists of codon sequences, which are units of 3 nucleotides that code for individual amino acids. And so the position of each of these 3 nucleotides in the right order is considered the reading frame. So here, for example, in the DNA, the reading frame consists of TAC, TGG, CTG, CTC, etc. Position of each of those in reference to the next is in the frame of the original positioning, the reading frame. And because they exist in groups of 3, we can see in a frame shift mutation that by adding or removing a nucleotide, we can actually shift the grouping of these bases and change the codes for the amino acids. So the main idea is that a reading frame can be shifted by adding or removing nucleotides, and as a result, we shift all of the amino acids resulting downstream from that position. So this is similar to a nonsense mutation in that it also results in a massive change in the protein, and almost always results in a lethal or non-functional protein. So I'll let you pause this and go look back at it. I've got another figure to sort of simplify this down here. But here you can see the original DNA sequence, and this little arrow is indicating that the G has been removed. And so this is what's referred to as a deletion mutation. So deletion, one of the 3 types of point mutations, will now shift all of the reading frames downstream from that over one position. And so as a result now, we'll see that the outcome of the amino acids downstream from that are all going to be changed. The same can occur with an insertion mutation. By adding an extra nucleotide, we can shift all of these downstream as well, and that will affect all of the amino acids as well. Let me show you a simplified way of looking at this here. Think of a basic first grade sentence like the dog ran. And I use this because we have 3 words with 3 letters each, just like codons. So you can think of each word as being a codon, each letter as being like a nucleotide, and the outcome is a coherent sentence. We can visualize what we mean by using 3 letters with the right words and 3 words with the right letters in the right position here. Now think of what happens if we insert an extra letter. Just the letter X could be any extra letter here. We insert a letter into the position right after the H here. And if we keep all of our letters in the same reading frame, we now have a nonsensical sentence here. This no longer makes any sense because we're trying to read it in units of 3. We've now added a letter that doesn't fit, and so now we have gra, and then in any letter that would occur in our sentence thereafter would be affected downstream from that. So this would be the idea of a insertion frame shift mutation. We insert a letter, we throw off the reading frame, no longer does our sentence make any sense to us. So if you go back here and try to look at this again, you'll see that this is also happening. We're inserting, or in the first one here, for example, in the first one here, we're removing, and as a result, we're throwing off the reading frame. Now this could also happen in reverse with a deletion. So we remove the E in this case, we shift everything over, and once again, we get a nonsensical sentence here. So that's the idea of a frame shift mutation. Because we're in sets of 3, by shifting through either adding or removing, our frame doesn't shift with it. The frame stays the same, but the nucleotides shift in reference to that frame. And as a result, the outcome is a sequence that makes no sense in reference to the original context. And so this is why you almost always get a nonsensical, non-functional protein in comparison to what you had to start with. So frame shift and nonsense mutations tend to be the most lethal, most damaging on the proteins themselves. Substitution can be harmful, but it depends on what exactly happens, because in some cases you may get a silent mutation with no effect. In other cases, you may get a nonsense mutation with a dramatic effect. So point mutations being substitution or variable, insertion and deletion are almost always harmful as they result in a frame shift mutation. You may have to go back over that to look at that again. Some of that stuff doesn't stick the first time you hear it, but go back over that, and we'll finish up here by wrapping up or summarizing the effects of mutation. So ultimately, as we talked about before, protein function is determined by shape. The shape of a protein is a consequence of the amino acid sequence. Any change in the amino acid sequence can result in changes to the protein shape. Should underline the word can there. Can result in changes to the protein shape. Changes in protein shape result in a loss or change in function. Changes can be subtle or dramatic, depending on exactly where that change takes place. And the outcome can either be harmful, neutral, or beneficial. Oftentimes it depends on what protein, how vital it is, and the environment around the organism itself. So the environment, like we saw in the peppered moth, or like I used in the example of body hair, depending on the environment and the organism itself, that can either be harmful, neutral, or beneficial. So that's a really quick overview of mutation there, looking at what it is, how it can occur, spontaneous or induced, how the environment plays a role, and then what are the specific ways that a point mutation can occur within the DNA and the outcomes with those point mutations. All right, so we'll wrap it up there. That'll be the end of our genetics lecture. And in the next section, we'll take a look at some of the ways genetics is used for biotechnology and for things like genetic engineering in Chapter 10. You guys have a great day.